So hello, everybody. Good afternoon here in Europe, and welcome to our EPIC Online Technology meeting on bioimaging. It's 13 February, so it's time to talk about bioimaging technologies and applications. It's a very exciting topic, so I'm very happy to moderate this meeting. Just a few words about EPIC. EPIC is the European Photonics Industry Consortium. It's an association of more than 800 corporate members. All of them are companies or research centers somehow related with photonics. And the goal of EPIC is to connect people, to ensure the sustainability of the photonics business in Europe, to give our members the opportunity of find new collaborations, new partners, and also to increase their business. And as I mentioned, today we have a very interesting meeting. We will talk about a lot of bioimaging related topics. So I'm very happy that we have this agenda with covering not only microscopy, that was my initial idea, but also optoacoustic imaging. We will have also some nanoscale sensing. We will have also LED illumination topics, optical nanoscope chip-based, OCT catheter imaging. So we are covering a bunch of different technologies that are related with the imaging of biological samples. So as I mentioned, I'm very happy to have all these topics and also to have this nice set of speakers from very relevant research centers and also top companies in this field. We will start with some presentations, and you see in the agenda, and then in the middle we will have some time for the sponsors presentation that we want to thank you for the support to the activities. Today the sponsors are the companies Actar Advanced Coatings, Chroma, Oxius, Iridian Spectral Technologies, and Humne Photonics. As I mentioned, I will introduce you later on, and you can have time to present your companies to talk a little bit about your activities. And as a summary of all the people interested in this topic, these are the supply chain imaging of all the companies registered to this event. And I'm also happy to see a lot of different companies, not only microscope manufacturers and components manufacturers, including optics, lasers, filters and coatings, the detectors and cameras, but also different biomedical imaging and biotechnology manufacturers, also end users, of course, and investors and consultants. That is very important for a lot of small companies, startups, spin-off, to know that it's a topic interesting for investors and consultants that will support their activities in the future. And that's all from my side here at the beginning. At the end, I will mention a few events that we will have in EPIC in the next months, but it's time to start with the presentations. We will start with the challenges into photon-excited microscopy and light sheet microscopy. So I will introduce you Pablo Loza. He's a staff scientist and SNA chief at the ICFO, the Institute of Photonic Science in Barcelona. So I will stop sharing my presentation. So Pablo, the floor is yours, and I'm very excited to see what you want to present us today.
Thanks, Antonio. Can you see my screen now?
I can see the screen. Perhaps you can put it in full screen mode.
Fantastic. Yeah, okay. Thank you, Antonio, for the introduction. And yeah, I am in the super resolution light microscopy and nanoscopy lab at ICFO, and I'm going to talk about challenges in two-photon microscopy and in light sheet microscopy. Just a few words about my activity. We have three pillars in my group. We do research and development, and then we adapt, merge, and develop novel instrumentation, for example, microscopy techniques. But we want to do this because we want to attract special users, users with a specific need. And also our third pillar is, of course, to train to the new generation of researchers with different backgrounds and according to their needs. So our activity runs in these three axes. And in terms of research and development, I have a number of microscopes. You can see them listed here. And of course, these are modified so that they have special capacities. We add new techniques, we add photonics instrumentation, we develop software, we develop a number of things so that we provide really unique systems trying to attack the most challenging pressing need in biomedical applications. At the same time that we do research, we also open our microscopes for users. And on the bottom part of the slide, you can see some numbers of the type of activity that we do as giving access. Okay, this talk is about light sheet microscopy and nonlinear microscopy. I will start with light sheet microscopy. Probably you know that in light sheet microscopy, we use a cylindrical lens not to focus the light into a point, but into a plane. That is why sometimes it is also called as selective plane illumination microscopy. One of the greatest things of this microscopy technique is that it is really fast for imaging in 2D. As comparison, in confocal microscopy, you have to scan one point until you have your image. And here you have this 2D image really, really fast. So the way it works is you have your light sheet, and then you pass your sample through the light sheet, and you have a second objective at 90 degrees where you collect your image. This is very important. Notice that for forming a 3D image, you need to move the sample through the light sheet. If you do that, then you can have images like these ones. This is a development of a zebrafish embryo where you can see a number of events happening there. We have also been using this technique to analyze the electrical activity on the zebrafish brain, in vivo zebrafish. And then from that, we can compute connectivity maps and then learn what are the circuitry in the brain of the zebrafish. Of course, this is really great, but the imaging speed is limited to approx 1 Hertz, 1 volume per second. And it would be great if we could capture even faster details than that. So how we do that? Well, there are several solutions, but the first thing is to move the light sheet, but not move the sample. So we have solved that problem somehow using either electrically tunable lenses and extended the depot field using face masks. This is a bit complicated. The sample is static and it can be maintained in health conditions. And the only thing that you move is the light sheet. This is great, but once that we have this, we have found several challenges. For example, if we use electrically tunable lenses, we can go up to 25 volumes per second. We can cover small volumes of our sample, and one volume is made of 10 planes. Just for you to have a comparison, in resonant confocal microscopy, you can go up to 30 Hertz. This is 30 times the same plane. And we can go here at 25, not planes, but volumes per second. What happened? If we want to go faster than 25 volumes per second, then the electrically tunable lens doesn't behave, and we lost linearity on its tunability, and we lost its amplitude. That means that we can image smaller and smaller volumes. So this already constitutes an important challenge in our research. What happens if we use wavefront coding by extending the depot field? Well, this technique has the potential to reach up to 100 volumes per second, which is, I think, is really nice. The problem is that it needs calibration through the measurement of a PSF, and this needs to be done for every sample. And once that you measure your PSF, then your image requires the convolution. That means that the images that you need to obtain need to be intrinsically with high signal-to-noise ratio. And that imposes several challenges. First of all, we need to have brighter fluorescent markers. If we have brighter fluorescent markers, the signal-to-noise ratio will be higher. This is one condition that we need. Second condition, we are driving our CCD cameras or SCMOS cameras as fast as they can go. Obviously, the fastest that you can go, the smaller is the photon budget. So one way to solve this problem is to do binning or to have larger pixels. But if we do that, we lose resolution. So we want high sensitivity, but also very high resolution. Third challenge that is really important is that if we are capturing very fast volumes, lots of 2D planes, in a few minutes, we will have lots and lots of data that needs to be stored, processed, and accessed. So these are some of the few challenges that we are facing just now in light sheet microscopy. for trying to get fast volumetric imaging. Now I am going to switch to nonlinear microscopy, and this is in the context of an European project that is Advanced Multimodal Photonics Laser Imaging, in which a number of research groups and companies are involved. So what's the challenge? In here we have the bladder and cancer bladder, and the idea is to identify cancer in the bladder in the first stages. You can see some numbers here, and of course, what we want to do is to detect the cancer in the initial stage. But bladder, the thickness of the bladder is 3 millimeters, so the light cannot penetrate all the way through the bladder. So what we do? Well, we have been exploring the different optical windows to have larger penetration depth inside the tissue. So normally we use the first window that is in the 800 nanometers. The second optical window has been explored, but the third optical window, which is around 1.7 microns, has been little or poorly explored. The nice thing is that if we stretch the bladder, this can get a thickness of around 1 millimeter, and at 1.7, working at 1.7, this is already accessible. The full thickness of the bladder stretched is accessible with optical techniques. So what we want to do, we want to explore nonlinear imaging. But nonlinear imaging, why? Because it has the opportunity to provide images in a label-free fashion. So the idea is then to use 1.7 microns to explore third harmonic generation, 3 photon excited fluorescent, 2 photon excited fluorescent, second harmonic generation, and of course elastic scattering. For accessing the 1.7 nanometer wavelength, normally we use OPO or OPAs. These are large systems and they are normally difficult to operate and they are expensive. Now, what else we want to do? If we frequency double that source, then we can access 850 nanometers, which is comparable with conventional TISAF wavelengths. And then we can do the usual nonlinear microscopy that we are used to do, 2 photon excited fluorescent, second harmonic generation, and so on. So in here, what are our challenges? We have the OPO, and of course, we really would like to have a compact ultra-short pulse laser at 1.7, and this laser should deliver pulses of around 100 to 200 femtosecond and have a repetition rate of around 10 to 100 megahertz to reach the high peak power that is needed. Other challenge is that we would like to be able to compensate the dispersion, because when the pulses travel through the different optical elements, they get larger, and then we lose intensity because we lose pulse duration. Another challenge, working at 1.7 is complicated because of the transmission of the objectives is not good. It's around 10 to 30% at these wavelengths. Where can I see other opportunities here? Well, it would be nice to have, I'm not expert on endoscopes, but it would be nice to have endoscopes with high transmission of 1.7 microns. Of course, this would have to be polar core to transmit ultra-short pulses. And these endoscopes should be very efficient in collecting any signal that is being generated with at 1.7. And of course, the last requirement is that fluorescent labels are not normally compatible with in vivo imaging for humans. So I see another opportunity there, trying to make fluorescent labels that are compatible for the human. And with that, I would like to thank you, and that's it.
So thank you very much, Pablo. Very interesting. Thank you also for sharing your challenges and opportunities. So it's time for questions. I encourage you to raise your hand so you can make your question directly to Pablo, and we have a rich discussion. I see a first question from DTU Health Technology. So please, the floor is yours.
Thank you very much. Thank you very much for a nice presentation. I have one little comment. You said there were no fluorescent labels available for in vivo use. I think you've completely overlooked a quite old technique called photodynamic diagnostics. Several of the endoscope companies like Storz, for example, have been using this for quite some years. It has its flaws, of course, because you have to administer this fluorescent label, but nevertheless, it can be done. especially for bladder cancer, but also for brain cancers, this technique have been used. So just a little piece of information for you.
Sure, no, that's why I put it as an opportunity. No, I'm not an expert on this type of markers. So thank you for your input.
You're welcome.
Thank you, Lars. And any other question for Pablo? I have one question regarding the comment you mentioned, one opportunity also for using endoscopes with hollow fibers. What's the idea behind that? Is that you use the endoscope to bring the light directly to the tissue, or is any other advantage of using the endoscope or the fiber?
No, for generating a nonlinear signal, you have to deliver the pulses into the sample. And in this case, if these are inside the human, you need to use fiber. And hollow core fibers are very good to transmit ultra short pulses through it. So again, I'm not an expert on endoscopes. That's why I put it as an opportunity. For me, from the perspective of microscopies, it would be really, really nice to be able to deliver the pulses in that position, but at the same time to capture any signal generated using the same fiber. So, yeah. The different fibers are used to send the ultra short pulses or to capture any signal that is being generated.
Yeah, so I have another question from Jessica from Edmuotics, please.
Hi, and thank you very much. It was a very interesting presentation. I do have just a quick clarification question. You mentioned some of the challenges, it being the transmission of high NA optics. Can you elaborate on specifically where the challenges are and how all of us that are optics manufacturers might be able to help you? Yeah,
the challenge that we are facing is that we have an OPO that is working at 1.7. Now, the OPO is a homemade system, and it delivers around 50 milliwatts average power from at the output. Now, the pulses have to go through the different optics of the microscope and then being focused by an objective. These objectives are normally from Nikon, Olympus, Zeiss, Leica, and so on. And these are not, the transmission of these objectives is not great at 1.7 microns. So the transmission that we have measured is around 10 to 30%. So we are losing already a lot of power there. So it's in this way, if you have better transmission on the objectives, you can relax the conditions needed on the laser for delivering the intensity that is needed to generate the nonlinear signal.
Okay, thank you, Pablo. I guess the question is more or less answered, Jessica. So we have another one.
Thank you.
Yeah, thank you. Alain, please.
Yes, I have 2 comments. Thanks for the talk. On the microscope, it reminds me when I started working at 1550 back in the 80s. I mean, that's 1980s, so you know I'm old. And what we did is that we took a standard microscope objective, send it to a coating, and then they dismantled it, removed the coatings, the AR coating, and then put a new AR coating. That increased significantly. That's the problem that you're probably facing, is that it's the coatings on the
correct.
And that's what we. And then now you can find objectives that are coated at 1550, but then they didn't exist. That's probably. The other thing is also, there's a company actually walking distance from here. It's called Kestar Optics, and they make double clad fiber couplers. So basically you can send the light in single mode and then capture the light in the cladding. So the NA of the captured light is really, really high. And they use it to do, you know, for example, OCT and confocal or something like that. I'm not a salesperson for that company, but the point is that they can collect the fluorescent light or the reflected light in the cladding, and the NA then is very high, like 0.22, 0.4, or something like that.
Okay.
So I think it may help in your endoscope. So that's all I have to say. No question, more comments. So thank you.
Okay. Thank you for your two comments. In terms of the coating, you are totally correct. One option would be to send all the different optics that are involved in an objective for coating. The thing is that we use our microscope in a very multimodal fashion. For example, for this same project, we have one laser at 800 that I have, one laser at 1030. We have one laser at 1.5 microns as well, and we have the OPO. So we are sending basically all these wavelengths through the same microscope to the same sample. So yeah, one option is to have dedicated optics for that. These are probably available if we send it to be made. But yeah, also it would be nice to have some optics that would be a little bit more less lossy at this range of wavelengths. And of course, we also use that microscope as a confocal. So we also have the visible wavelengths inside there.
Yeah.
But yeah, there are solutions. I know that there are solutions, but yeah, at least we're facing with this one. And in terms of the cladding of the, it was a fiber, right?
Yeah.
Okay.
It's a double flat fiber.
Yeah.
I'll send you the link in the chat if you don't know the company.
Thank you very much. That would be really, really useful. Using fibers with ultra-short pulses need to be used together with a very nice system for dispersion compensation. But this can be done and can be explored. But I mean, a hollow core fiber would solve the problem really neatly.
I have another question, if I may, very quickly. Have you considered using fiber lasers at 1700, either thulium dope or Raman shifted? And also, I would recommend that you consider a little bit longer pulse, because people always forget the integral when they look at the signal. Yes, it depends on the peak power, but in tissue, you're limited in peak power and average power. So if you drop the rep rate and elongate the pulse, you will get roughly the same signal, but you will suffer much less from dispersion.
Correct. Yeah, thank you for the
comments. Talk at Photonics West by Uber and everything, and he even went to nanosecond. And people always forget the integral.
Yeah. No. No. Thank you for your comment. You are spot on. In the project that I have presented, Amplitude, we have two teams dedicated for making compact ultra-short lasers on the techniques that you have mentioned. So we are on it, and hopefully in the next months we will have some compact system available to us that it would be nice to compare to other commercial systems. And in terms of the peak power, this is totally correct. Normally use the workhorse laser system is the TISAF. The TISAF works at around 80 to 100 megahertz. If you work not at 100 megahertz, but at 10 megahertz, which is something that we are exploring, then you are also increasing the peak power by an order of magnitude. Instead of working with 10 femtosecond pulses, you reduce the frequency of repetition to 10 megahertz, and it gives the same peak power as if you had 10 femtosecond pulses. The advantage being is that 10 femtosecond pulses are really, really high to compensate for dispersion, but 100 femtosecond pulses are not. So this is an approach. Reduce the average power. Now the other thing is, if you reduce the average power, the dwell time, so the time that the pulses are in 1 pixel, is less. So it comes with some pros and cons.
Yeah. But consider picosecond pulse as well, or even Uber demonstrated even nanosecond pulse. So anyway, all right, thank you.
Thank you, Elena. Thank you, Pablo. We will have time at the end to continue the discussion if there is something pending. So if there is any other question, please keep it the idea that we can discuss at the end. And Pablo, thank you very much for your nice presentation and for the discussion. I see that everybody was really interested. You also presented about, you talk about fluorescence, and the fluorescence is important filters, and we have the company Chroma that are manufacturer of filters, and they want to share with us one slide showing the capabilities. So Almut, if you can share your screen and unmute yourself, you can present your slide now. We see your slide, but we don't hear you.
Okay. So okay. I hope you can hear me now. Thank you for giving me the opportunity to present.
Sorry, Almut. We have here.
I guess it's. I'm supposed
Patrick.
to. Yeah.
Yeah, but can you stop sharing? I will introduce you later on. It's just the time to Almut for one slide, and you can start later on.
Okay, so you
Almut,
see,
you can share again.
You see my slide or no? Okay.
Now we can see it. So if
now you can see it. Okay.
Yeah.
The full mode. You can see the full mode.
No. You have to go down. Yeah.
Okay. Now it should be full mode.
Yeah.
Yes. Thank you for giving me the opportunity to say a few words about chroma technology. So we're producing optical filters and dichroics. We're doing this since 1991. So we have a great experience and wide range of filters that we make. We make filters and, of course, dichroics in various sizes, and we use sputter coating technologies, state of the art, and our spectral range is from around 200 nanometers to 3 microns. We make parts, filters, dichroics, as I said, in various sizes for various applications. Fluorescence is the applications that we started with. So we have a really wide range of parts in our catalog that are for fluorescence microscopy, but also for flow, for Raman, for astronomy, for PCR machines, light sheet. You name the application. I think we can find something or make already something for this application. We offer small batches. We offer high volumes. We offer small batches with R&D and design service, so that we make the filters that exactly match your application best. That's what it says on the right side here. So we also do rapid prototyping so that you can start with a small batch of parts and test them before you actually go into a volume production. And I think some of our customers are in the auditory, so you know our service and how we perform and how we act. So thank you for giving me the short option.
You can contact Almu and Chroma in case you are interested in any of the products, or contact me if you want to be connected with them, and I will be happy to introduce you or they can explain all the catalogs. And we can go now with the second speakers. So sorry for interrupting you, Pratik. You can share your screen now.
No problem. I was just not aware that there's a between.
No problem at all. So we are moving from microscopy to optoacoustic imaging. Very interesting technology, and I'm very excited to hear about what is happening in Ithera Medical. So we have Patrick Leishin, CTO from this company. The floor is yours.
Okay, hello everybody, and thanks for Antonio for inviting and introducing me. Just a question. Can you see my screen?
Yeah.
Okay, so this is about Ithera Medical and optoacoustic imaging. Antonio asked me to give you an introduction of what we are doing and why we are doing it, and what we do. What do we need from the optophotonic community? So my name is Patrick Leishin. I'm the CTO from Ithera Medical, and this company is around for quite a while, 12 years. And what you see is here on the left-hand side, the device. So it is clearly a medical device, and this is quite advanced. As you see, a doctor is using it in front of a patient, though it has a CE label. So what is left right now, it's here the next step. What we want to do, we want to transform this device here into a device for routine clinical use. And we need better optical components, better lasers. We need FDA approved and clinical trials process. So technology, just one slide. And I guess the thing I want you to remember is light in, sound out. So how can it be that you can really get microscopic images from some centimeter tissue in the skin it is so if you excite with a laser light the tissue here, you can penetrate quite some centimeters. Then the only obstacle is the light would not get out, but if you have a high energy laser like 10 millijoule and excite here with short laser pulses like 10 nanoseconds in 3 centimeters, you will generate ultrasonic waves. The molecules start vibrating, and this is generally generating the ultrasound waves. And these ultrasound waves will penetrate the way back through the tissue and can be detected by a standard ultrasonic detector. So what do you get then? You can get an ultrasonic image like any ultrasonic device, but if you tune the wavelengths, you can get a molecular image. For example, you could image at 650, at 800, at 1000 or 1300 nanometer and measure the absorption of the molecules in 3 meter depths, for example. And easily you will get an overlay of the ultrasonic image with some molecular images. For example, you can detect blood, whereas blood, a tumor in a breast, for example, has a very high blood content. So you easily can see this object is a tumor or is an emboli or whatever. So technology-wise, light in, ultrasound out. What you need, obviously, is then a laser and fiber delivery. Just a historic view on this imaging devices technologies here to show you the dimension of what we are after. In the early 90s, we had the ultrasound. Then in the 70s, we got the CT and the MRI. But not only get we anatomical information, but functional information. And then 80s, 90s, and now 2000, we see that more and more optical technologies can get you molecular pictures, images. The obstacle for all these technologies is if you look for the year of commercialization and the installed base, it takes 6 years or so from the first medical device until it's really in clinical use. So 6 years is quite a while if you are depending on venture capital money. However, the markets are big. We are talking billions here, and even the small amount Itera is after would be good enough for 150 in 2030. So where are we today? What is the status as of today? I already showed you we have a device. The device has a CE label, very important for any clinical trial. Plus, we have a large IP portfolio, very important also if you want to have a competitive edge. We have done some medical studies, and we have here a lot of key opinion leaders, so be it the Technical University of Munich or the Johns Hopkins University. Almost every important university here has some of our systems, be it preclinical or clinical systems. Revenue-wise, we already sold about 40 million in total. So this is not only about burning venture capital, but we have a solid revenue over the past years, and we also are engaged in research projects. So competitors, you might be curious, who are competitors? Yes, there are two types of competitors. We have the optoacoustic specialists, which is, for example, Xenu or Luxenos. Xenu from US is concentrating on breast imaging. Luxenos is also concentrating on breast imaging, but they are not active in Germany and in Europe. The big ones here, like Siemens, Healthineers, Philips, and GE, they are closely watching and observing what we are doing, but do not have an active R&D development program. So what do we look for? I guess it's good to see where we come from. This is the past. We were using complex and bulky OPO laser systems. They did the job, that's fine. But they had a very, very low wall plug efficiency, which is good for a research project, but it's not good for clinical use. We own 20 patent families. We got the German Innovation Prize and 2019 the CE mark, and this opened the door for real clinical trials. So what we are doing right now, we scale and want to gain leadership in the maturing market. We have a compact OPO laser system integrated right now with a fiber array for delivery. We are performing FDA and clinical trials, and we want to grow the revenue for the clinical part to beyond 10 million in 25. What happens 25 and over, beyond? you see we want to prepare the wide the clinical routine use market. And here we will need diode-based laser systems with optimized bundled fiber arrays for delivery. We will make this for routine clinical use, means that not only a radiology department can use the machine, but maybe any doctor in any hospital. And we expect that the revenue will then 2030 or so be close to 100 million euro. In more detail, what do we need? A laser system. Laser system as of today is tunable between 650 and 1300 nanometer. We have 10 millijoule average power, 10 nanoseconds, 25 to 100 hertz, and the power consumption should be lower than 300 watt. The fiber delivery today, it is a circular entry. We use 300 fibers, which transform then to a flat top output in a handheld, and the transport efficiency is about 70%. And if you want to have a look for a drawing, this is how it looks like. It's a circular sport here, and it's a flat top output in here. So, and any company which is interested in doing joint development and research projects is more than welcome. So thanks again for the time having me, and any question by email, by phone, or now, please go ahead.
Thank you, Patrick. Very interesting, and thank you for joining also with us your needs and your future ideas. So it's time for questions. I have a question for you. It's regarding the markets or the applications that you are going to focus with your device. You're going to go in the oncology related topics, skin treatments, systemology. Which applications do you have in mind at the beginning?
So the interesting question is what you are after. There are 30 applications where optoacoustics is useful. What we have in mind is a general purpose optoacoustic machine. So for the first FDA submission, we are not concentrating on one treatment, on one application, but we say, well, we have a machine which can detect a lot, and the doctor himself or herself needs to make a treatment decision. But you're absolutely right. You could do a treatment monitoring, for example, oncology, breast cancer. After one treatment, you could see if the tumor is responding to the treatment. You could go for a skin cancer, or you could even go for deeper tissue problems, NMD, for example. This is all open. And the big question is, what brings the most money? Sino Medical went for breast cancer. Okay, we prefer to go rather for fibrosis. There are things where no treatment is available today from a strategy point of view, and the doctors would more than welcome our device, and it's not so complicated in the diagnosis.
Okay.
But it is all open, 13 potential. We have made our selection to a wider range of applications.
Thank you, Patrick. We have another question from Regina Gumeniuk from Tampere University.
Hello, thank you very much for interesting presentation. First of all, one comment regarding fiber development. I recommend you to contact Ceram Optics. They're specializing, especially for medical application. And if you need assistance, I'm happy to help because we collaborating a lot with them.
Yeah. Thanks a lot. It could be so that Ceram Optic is already our supplier or one of the two suppliers.
Great. The second question regarding the laser, you mentioned quite broad wavelength range. Do you need tunable, or it can be any wavelength within that range?
It depends on what you're after. If you know what application you want to have, for example, breast cancer, this is why Seno concentrated, I guess, 850 and 1060, easy wavelengths, not tunable. But in our case, as we want to have a general optoacoustic device, this is tunable, and very, very often we use 4 different wavelengths depending on the application. So if you want to monitor blood, if you want to monitor nerves, if you want to monitor tissue or so, typically it comes down to the fact that you start 650, 70, 700 something, 850 and 1100 or so. But if you nail it down to the one or the other application, two laser diodes or 2 wavelengths should be okay. And if you come from a research point of view, like IBMI here in Munich or so, to get the best and fantastic image, you can imagine you would need 64 wavelengths.
64.
64. Well, yeah, this is, for example, what is state of the art in people doing artificial intelligence-based deep models here. But this can not be done in an online way, but in post-processing only.
Okay,
so
thank you very much.
The more wavelengths you add, for example, if you have a 25 Hertz repetition rate, the more wavelengths you have, the less repetition rate you have, and the more calculation power for the images you need. So
okay.
Research-based, I would say 64, state-of-the-art, more would not help, obviously. But for a clinical application, for a real clinical application with a high volume, 2 wavelengths would be enough.
Okay, thank you very much.
Thank you, Regina, for the question. Yeah, Simon from Lazy 2000, nice to see you.
Yes, I have another question concerning the different wavelengths. Do you need shot-by-shot tunability?
Yes and no. Well, right now we have systems which have shot-by-shot tunability, but we have other systems where this is not needed. You would average typically, but sometimes shot-by-shot in some research systems we have the shot-by-shot.
Okay.
It's then not an OPO, it's a Raman, tunable Raman. But there is no yes and no answer, depends a little bit on the application. But in general, for a clinical device, shot-by-shot is not needed.
Okay, maybe we should talk after the meeting, so go into details.
Yeah, of
because
course,
we
just
have
give me a
something for
call
you.
or send me an email.
Yeah.
Perfect. So yeah, let's continue the discussion after the meeting. If you need help to connect, just let me know and I can introduce you both. You can continue the discussion. Thank you, Patrick. Very interesting presentation. A really nice device, and I wish you the best for the next steps. And let's move to our next speaker. Is Jana Nieder, the head of Nanophotonics and Bioimaging Corporate Laboratories at INL in Braga. And she's going to talk a little bit about the amount of systems they have in their facilities and their expertise with bioimaging. So, Jana, the floor is yours. And thank you for being here today.
Thank you very much for the invitation. It's a pleasure to present in this panel. And I will present some with the 2 different heads, right? One's as the head of Nanophotonics and Bioimaging Core Facilities at INL, and on the other hand, as the research group leader of the ultra-fast bio-nanophotonics research group, where we develop functional imaging solutions that are characterized either by nanoscale sensitivity or bioenergetic imaging. To introduce you to INL, this is an international organization that was founded by Portugal and Spain, and it's focused on nanotechnology, aiming to become a recognized leading global nanotechnology innovation hub, helping to innovate in nanotechnologies for to reach sustainable development goals in various areas. And the way we do that is by exploring interfaces. Interfaces meaning interfaces between different research areas, between material science, biology, life science, and computation. And at the moment, as I mentioned, we focus our efforts in 6 different research clusters: nanotechnology for food, for precise personalized health technology, clean energy, sustainable environment, smart digital nanosystems, and advanced materials and computing. There are 24 research groups working in these topics, and at the moment around 426 researchers, as well as administration and grant office from 37 different countries. The facilities that we have are open access facilities, so external users can use it either from academia or from industry. And we have a different set of facilities, including the nanophotonics and bioimaging, of course, but also one of the largest clean room facilities in the in Portugal, and with standing out 9-inch wafer micro nano fabrication facilities that allow to have prototyping, but also small scale pilot line fabrication. than advanced electron microscopy and SARS XPS techniques. On the microscopies, we have on the one hand side commercial systems that you can see here, and I was asked to present a little bit about the experience, how it is to operate, for example, a confocal Raman microscope in a user access facility. And indeed, in our health cluster, where it is to develop precise personalized health tech technologies, we successfully provide these microscopies to the user base. And here you can see an example of nanostars that were generated to be SIRS-based single-cell multiplex phenotyping sensors, such that they bind to very selected proteins at the cell surface and are able to be used in cancer research, combined with some microfluidics technology where single cells are separated in single droplets, and the SIRS sensor is successfully used to identify different cancer phenotypes. Besides the commercial systems that we use as they are, we also develop our own microscopes, and I wanted to highlight some of the multiphoton, second harmonic generation, and fluorescence lifetime imaging microscopes, as well as the combination of scanning AFM with confocal spinning disk microscopy, as well as some advanced spectral imaging ellipsometry by my colleague Peter de Boyle. And one of the other highlights that may be different from other bioimaging core facilities is that we often combine the nanofabrication with microfabrication with microscopy. So what you see here is a 2-photon polymerization workstation, in this case a commercial one, but in the meantime we also develop our own 2-photon polymerization setups that we use to, for example, construct 3D scaffolds in which cells can be grown in 3D models. And ideally, we are part of providing advanced 3D disease models that can be used for in vitro studies of diseases or to test novel personalized treatments. One of the interesting parts to test whether the cells are alive is, of course, something that we call cellular bioenergetic imaging. And here I wanted to mention some of the opportunities that we see using also ultra-short laser pulses. Here, an example where we use a conventional titan sapphire laser to test the interaction of cells, cancer cells, with the drug delivery system, and where we mimic where we test the metabolic state of the cells by measuring fluorescence lifetime images following metabolites such as NADPH compounds that have a different fluorescence lifetime when they are in a bound or unbound state. But we want to push this further, and that is one of the ongoing research projects where we work with in collaboration with the company Sphere Ultrafast Photonics from Porto, that is providing and developing an ultra-short laser pulse. So here we don't speak about this mentioned 80 to 100 femtosecond, but down to 7 femtoseconds laser. And you can see the spectrum here on the top, where we have more than 400 nanometers spanning ultra broadband excitation light that can be used to excite many chromophores at the same time in a sample. And that can also be used to reach deep into tissue. And here you can see a video of a recently imaged spheroid where we grow spheroid samples and image them at deeper tissue layers compared to what we're able to do in a conventional 3D confocal microscope. The main focus of the research team is in advanced bioimaging and sensing is manifold, from functional imaging, extracting physical parameters like temperature, magnetic field out of samples, using nanothermosensors, for example, to using this combination of nanotechnology for near field sensing or microtechnology for building 3D scaffolds combined with few cycle lasers for advanced imaging of those. We also use related technologies like quantum photonics, where the quantum principle lies in doing magnetometry based on detecting also fluorescent signals, but in this case out of single defect centers in diamond to measure magnetic fields. And we are curious how to integrate these technologies with what we do at INL in photonic integrated devices, where we have several European projects ongoing at the moment. Chip AI just came to an end, and Insect Neural Nano is a new EAC Pathfinder project coordinated by Bruno Romeda in my research team that is developing neuromorphic, bio-inspired spiking nano light sources. With this, I would like to thank you for the opportunity to speak about the ongoing research and some of the challenges. For the challenges, we think that the amount of data collected in the 3D life cell models and 3D disease models, we may need to include machine learning approaches and advanced data analytics, and certainly to bring some of these microscopies into small, cheaper and more versatile mobile spaces. Some of the integrated light sources could be of benefit, as well as nano detectors that could move from a bulk microscope into an integrated solution for microscopy. And with this, I would like to thank and close, and I'm happy to receive your questions.
Thank you very much, Jana. Really nice presentation. I really enjoyed the results and your collaborations. So we have time for questions to Jana. I have one curiosity, and it's related to your collaboration with the Sphere Photonics, because I know them from the diagnosis tools for ultrafast lasers, and you mentioned that you are using their solution also to have lower pulse width, right? Do they have any kind of compressor for ultrafast lasers, or what was the technology?
Yes. Thank you for the question, and it relates, of course, to the discussion that we had in the beginning. Indeed, Sphere Photonics provides also the technology for ultrafast laser characterization and for the pulse compression, and we use that by we're involved in developing a prototype that enables us to characterize and compress the few cycle laser in the focus of the microscope objective. So we use the D-scan technology, but adapted to allow us to characterize the few cycle laser there. The technology is called D-scan, and
Okay, thank you very much. I saw Alexander, you raised your hand. Alexander from Toptica, do you still have a question?
Yeah, just indeed at that moment you asked my question. But I have another one, if I may.
Yeah, please.
What wavelength do you use for the two-photon polymerization for creating the scaffolds?
Yeah, in terms of material, or which type of setup?
Like a laser wavelength. Well, I mean, it may be adapted to the material, of course. Yeah.
Yes. So indeed, we work with different type of materials. So there's one that we actually get from EALS Force, which is called SC2080. It's a photoresist that is biocompatible, and for the fabrication, we typically work at 790 nanometer wavelengths. And more recently, we fabricate two photon polymerized materials, not only for bioimaging application or bioscaffolding applications, but also for optical applications. And there we work mostly with ormocomp materials, where we have to tune the wavelengths to lower wavelengths to 740, 730 nanometers. But yeah, this is always basically in the regime where the polymer is absorbing better. So it depends on the material that you use.
So, I mean, that's indeed interesting, because the other application which you do is NADH excitation for metabolic imaging, right? So both applications, if you now think about shortening the wavelength, both applications will benefit potentially from fiber lasers which go shorter, right? So if you have something like 740 in mind or so.
Yeah, so in this specific case, actually for the two photon polymerization, we typically use a standard tsunami, a tunable spectral physics, titan sapphire laser, and we have no problem to shift it down to 730 or 700 nanometers. we typically use a few tens of milliwatts at the focus of the objective, and operate at 80 megahertz. So I'm not sure whether the fiber lasers would provide the same power density, etc. So basically we have to optimize the fabrication process for each material. So typically it involves the scanning speed, the laser power, etc., which may also have an effect on the exact 3D voxel shape, right, that you use when you change the laser power at which you write. So it's a quite demanding process to have the optimization established. So you need to rely on a quite stable laser operation and to have reproducible structures that match exactly the 3D model that you design in a CAD program, possibly beforehand.
Thank you.
Another question from Elizabeth from Photonics.
Hi, thanks, Jana. Very cool lab you have there. Lots of very nice equipment. Just following on from Alexander's question there as well about on the 2-photon polymerization, do you see any advantage in going to even shorter femtosecond pulses? So like 50 femtoseconds from, I guess you're at 100 or 200 femtoseconds with the TISAP?
Yeah, we are at around 100 femtoseconds after optimization. Actually, as we were optimizing our lasers in the focus of the objective, we work now mostly with 70 down to the fully compressed pulse, 70 femtoseconds. But to test whether such a few cycle laser and really short pulses would make a difference in the fabrication is still to be explored and interesting. So we haven't done that yet, but I think there might be an opportunity to achieve ultimately higher resolution in the writing of the structures, as the peak density is very high and concentrated, and basically, I think that one could optimize it for smaller resolution. But again, depending on the application, that might not be needed. So for the 3D cell scaffolds, I wouldn't make the effort to optimize writing on below subdiffraction limited scales. So, yeah.
Okay, thanks.
Thank you.
Thank you, Elizabeth. And thank you, Jana, for very nice presentation.
I think just if there is an expert in the optical materials, polymers that indeed are superior in optical transmission, that could be really interesting to know for the optical interconnects and optical structures that we are fabricating. There's surely always a limitation.
Good. So
thank you.
Yeah, if anybody in the room is interested in discussing with Jana, please let me know so you can continue the discussion by email later on. And now, before going to the sponsors, we talk a lot about microscopy, and there is a technology most of you are aware that adaptive optics is coming from astronomy, but it can be also used for bioimaging applications. And we have here in the room Stefan from Phaseform. Really nice to see you, and I would like to hear what do you have in terms of adaptive optics for this kind of application. So if you can share your screen.
Yes, thank you so much, Antonio, for inviting me. What I'm showing you just briefly in a minute is a new class of optical devices, of optical components that are combining what you probably know from deformable mirrors with liquid crystal modulators. So we have a compact and transmissive refractive component that is just a tiny piece of glass, and there's a little liquid, and it's encapsulated by a membrane. And what that can do is actually change the phase of the light that is progressing through it. So it's like a deformable mirror, but transmissive. That means you can put it anywhere easily in a home-built microscope, or even you can just stick it between the turret mount, for example. And what we did so far with our startup phase form and our first product that we have on the market right now, we call it Delta 7. The transmissive continuous sheet wavefront modulator is an example from 2-photon microscopy, where we're showing here a single neuron imaging that you can improve if you're going deep into the tissue and correct for the aberrations, for example, spherical aberration that you encounter deep into the tissue. And I'm happy during the course of the discussion today, hear some alternative and new applications for the technology. I'm just giving here a few examples easily for subscale microscopes, or as also put it in as a camera add-on, for example, to commercial microscope. So there are plenty of ways of using the technology, stacking multiple of them in different directions, and wanted to introduce that new component into the fray, into the field as a new toy for the optics playground. And I think bioimaging and deep tissue microscopy are particularly prone for benefiting from this technology.
Very good, very interesting technology, Stefan. So as I mentioned, in the case of Chroma, if anybody's interested in more information or in further discussion with Stefan, please contact him directly or let us know to put you in contact with him to discuss your application. And well, Stefan, I'm sure that there will be another more presentations that will be interesting for you. So feel free to make your questions or directly to the speakers.
Thank you.
And yeah, you're welcome. And as I mentioned in the agenda, we are moving, in this case, the sponsors' time to the middle. So we have time for them. Thank you for your support. I would like to start inviting Actor Advanced Coatings. I see Alexander here. So please, you can say a few words about your company, perhaps share any slide. So it's your time.
Thank you, Antonio, for your introduction. I hope you can hear me well.
Yeah, we can.
I am going to share one slide. Very shortly and very briefly. I am the CEO of ACTA's German subsidiary. We are a company specialized in black coatings and coated foils, and we supply our coatings to numerous photonic and optics applications. There are three main applications. Suppression of stray light and scattered light in optical systems is by far the most important one, but we can also absorb laser power with our stable coatings and create high emissivity surfaces for infrared applications. And these coatings are also used in medical and bioimaging applications. They are inorganic and they have zero autofluorescence, so they can be used in fluorescence imaging. And we apply also coating to like lens housings of endoscopes. So we are also able to very precisely apply the coating in lens housings and optical elements for endoscopes. Yeah, that's it from my side. And thank you for the opportunity to very briefly introduce what we do.
Thank you, Alexander. Thank you also for your support in this meeting. I hope you are also enjoying the presentations today. So we go with the next sponsor is Hamner Photonics. We have Elizabeth Ely, head of marketing of Hamner Photonics. So Elizabeth, you can share your screen and tell us a little bit about your company.
Thanks, Antonio. Let's see, I will share my screen.
Yeah, you see the screen. You can put in full mode.
There, how's that?
Good.
Okay, My name is Elizabeth Illy. I'm the head of marketing for Hübner Photonics. At Hübner Photonics, we make high-performance lasers for analytical instrumentation. Hübner Photonics is a German company, actually formed only 5 years ago, but comprises largely of cobalt lasers, and cobalt is 22 years old. So 5 years ago, Hübner bought cobalt lasers. So that is a large part of our portfolio now. We have 4 product lines, the cobalt product line, and there we have CW and diode lasers from the UV into the near infrared. And we also have a multi-line laser with 4 of those lasers in one box, basically making it easier for users to hook up to their microscopes. We also have cobalt nanosecond lasers. Patrick, I know we talked at Photonics West, but maybe something of interest for you. So these are very small, compact microjoule per pulse lasers at 532 and 1064 for optoacoustics and LIBS. And then we have the C-Flex laser combiners, which is basically any of the cobalt lasers all in one box, again, making it easier. And then last but not least, a year ago, we acquired Valo Innovations. And now we have the Valo femtosecond series lasers, and they basically have less than 50 femtosecond pulse width. So, Jana, that was a little bit along my line of questioning to you if you see any advantage in going to even shorter femtosecond pulses, because basically from what we have seen and what we have worked with researchers at the moment, going to 50 femtosecond pulses basically increases your peak power, and then you basically need to expose with lower average power, which means that it's basically gentler on the samples and you can image for a long period of time. So if anyone is interested in trying to verify if this is actually true, please let us know. We have Oliver also sitting in the audience. He's an expert in this area. So that's basically, we're in the art of making lasers, high performance lasers for bioimaging and other analytical instrumentations. Thanks.
Thank you very much, Elizabeth, and thank you to Hübner for the support to this event also. Chroma is another sponsor of this event. So Almut, you mentioned a little bit about your optical filters, but perhaps you can say a few words more about your company. So you can share the screen. Or just say a few words as you want.
Well, yes, I think I can reopen. I just closed it because I thought I was done with sharing my screen. Yeah, Chroma was founded in 1991, so we're in the fluorescence market and in the optical market since many years now. And what I would like to point out in this audience is that we're very open to custom designs, to R&D projects, where you tell us what you would like to do, if you would like to reflect, to block, to transmit a certain wavelength. And as I said before, our comfort zone is 200 to 3 microns. So yeah, just let us know what you plan to do and we can try to find a solution. Our designers and R&D experts are always open and happy to work with you. And our sales team is known for quick and good support. So yeah, feel free to contact me. Antonio has my contact data.
Thank you, Almut. Thank you to Chroma. And our next sponsor is Iridian Spectral Technologies. I see that Hong Bai Lau, the sales account manager, is in the room. Do you want to say a few words?
Hello.
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 Yeah. Hear you?
Yeah. Can you hear me?
Yeah, we can hear you.
Yeah. I'm Hong Bai Lau from Iridian Spectral Technologies. We are the company just make optical filters. Our company founded in 1998 in Canada. So we basically make various optical filters for different applications, including this biomedical applications. We we make a lot of custom filters based on your customer's specification and requirements. We can optimize the filters for basically for fluorescence, higher signals and lower noise filters. So if you have any requirements for the optical filters, we can always work closely with you. So spectrum range, we are working on from 300 nanometer up to 15 micron. So that's but I don't have this slide to show here from my screen.
No problem, Hongbai. If there is any request, perhaps we can put you in contact with you, and you can discuss the needs of the customer. And thank you again for your support in the meeting today.
Thank you.
And our last sponsor is Oxius. I don't see Oxius in the room, but I can introduce them. It's a laser design and manufacturing house. They founded in 2002 to make special lasers in the visible range. The advanced continuous wave laser modules with different applications like biophotonics, metrology, spectroscopy, and other analytics and instrumentation. So if you are interested, please let us know. If I'm right, in Germany, Laser 2000 is the distributor, so you can also, you are in Germany, contact them. And thank you for your support to this event. And let's continue with the next presentation. Let's go with a very interesting custom microscopy talk. Our next speaker is Thierry Emerot, is Business Development Director at Lambda X. And yeah, I'm very curious about your presentation to see what's your developments for bioimaging. So the floor is yours.
Okay, thank you very much, Antonio. Can you see my presentation full screen?
Yeah, we can.
Great. So thank you for the opportunity to introduce our contribution to this meeting. And I'm Thierry Emerot. I'm a business development director at Lambda X. And as a short introduction, I think it's interesting. Maybe we'll have the possibility to look at this bioimaging more in an engineering perspective than a fundamental science perspective, as was presented by the former speakers. So outline of my talk, I will shortly introduce who is Lambda X, who we are and what we do, just in one slide. Then I will go through three different use cases that we have contributed to in bioimaging, and then we will go through the traditional epic questions. So Lambda X, we are an engineering and providing engineering services and OEM manufacturing services for custom optical systems. We serve different markets, as you can see here. However, today we focus more on the medtech and the life science market. We are close to 50 people, and we have contributed to 300 product development projects so far. And I would say our key contribution can be summarized this way: explore widely, I would say, different optical concepts and also engineering solution at whatever step of a product development stage, and co-create with our customer and globally de-risk product development projects at our customers. So now going through the different use cases. So the first one is a custom microscope that we contributed to make robust and reliable, and which is used for enzymatic quality control and process control. So our customer has developed biopolymer microstructure deposited on silicon plates, which they interacting with some materials which is used in different processes using enzymes, and could be in the food industry, could be in the detergent industry. And what we do and what they do in the end is, as you can see on the top right, is to analyze the shape and the volume of the modified biopolymer structures after their interaction with the material to test or the process to test. And from then on, they get a qualitative information on how big is the enzymatic activity. So you can see some pictures here showing the microscope, and what we have been doing is to make an initial design which was quite unstable to make it robust, reliable, with no instrument-to-instrument variation. We had a design to cost contribution to by making and developing a custom autofocus system. And today we are a partner for this company to manufacture in quantity 10 to 50 units per year. The second use case is about mid-infrared spectrometry and imaging on biopsies. And here we work for a company which is called Admir, which is a spin-off from Cevatec in Grenoble in France. So what is the context of the application? It is basically oncology. There are more and more tests to be performed on a growing number of patients. However, we face some shortage in anatomopathologists, and which results in a very, very small fraction of the biopsies which are currently read. So the idea of Admir was to propose an automated fast infrared imaging, and you could see it as bringing a well-known lab technology, which is infrared fast Fourier transform infrared spectrometry into a very fast device by combining three key technology bricks. First is the high brilliance infrared lasers, a bolometric matrix as a sensor, and bringing some artificial intelligence for image analysis. We can see some results here. So you can see on the top left, the traditional staining image you will see from a traditional biopsy. And you see that with the type of technology, you can get similar images. However, on top of this, you can make some tissue classification using this artificial intelligence software contribution. And what have we, and you can see also on the bottom left, the demonstrator, which has been built by Admir and already tested in some marketplaces. So what we have done here, we have made the original concept, and we have designed and manufactured a custom illumination box for that microscope, which is made of 8 multiplexed quantum cascade laser sources operating in the range 5 to 11 micrometer. And the challenge for us was to deliver an homogeneous illumination, and particularly managing speckles from those Korean sources, and reach better than 10% homogeneity over several square millimeter area. And on the other end, to reach a high enough signal over noise ratio to enable the fast measurement. As I mentioned, the target is to perform the images in less than a minute. So the third use case I'd like to introduce is different. It's about cell therapy, and especially one key step in this cell therapy process, which is cell expansion. After the cells have been transfected, you need to monitor how they develop and how they are transforming after the transfection over time. So here we talk about another kind of microscope, which is a digital holographic microscope, which is performing the online monitoring of the cell population all throughout the process and online. That means you can really monitor the morphology of the cell by label-free 3D live imaging. So as you can see here, when you start, you get some kind of uniform cell shape or morphology, but as time goes, after the transfection, the cell try to change, and actually the technology enables to discriminate between an infected cell, a dead cell, and a viable cell. And in the end, you can make a clear cell population diagnostic, which tells the pathologist or to decide what you should do with the material in the bioreactor. So here, what LambdaX has done is the industrialization of the microscope to reach full GMP and UL compliancy, because the major customer was in the US. And today we are also an OEM manufacturing partner to the Ovidio company. And we have built over 200 units over the past 3 years. So that was the 3 examples I'd like to highlight how engineering can contribute to bioimaging applications. And to end up my talk, we could go shortly through the EPIC questions. So what can we do for you? We would like to be your partner to innovate with optics for your products, for your whatever device. And we can do that as a matter. We like to elaborate for new product concept or to de-risk product development as a whole. And what could you do for us? We are eager always to work on new bioimaging application with optics in a very general perspective. And we could contribute to test or to integrate innovative components for such application. And well, maybe those will trigger some questions from the audience, but I would like to thank you for your attention, and I would be glad to answer whatever question you might have.
Thank you very much, Thierry, for showing some of your capabilities and also proposals to collaborate with other companies. Any question in the room? Yeah, Alexander from Toptica.
Yes, just if I noted correctly that you use 8 multiplex quantum cascade lasers to achieve the performance for the device. Is that correct?
Sorry, could you repeat the question? I didn't get it at the beginning.
I made a note that you use 8 multiplexed quantum cascade lasers, right? So my obvious question is if this should be potentially better to have maybe one source which covers at least this wider range that should make the setup potentially better. And maybe in other words, what other problems are you facing with the such a big amount of quantum cascade lasers?
Well, actually, the customer decided to do it this way to get fast, because they selected the 8 different quantum cascade lasers because they were emitting at the wavelength of interest, and depending on the application and the type of molecules you want to probe, they will change those sources. But they say that, well, in a whole spectrum or in a distributed spectrum, there are wavelengths we are more of interest, because you will probe the appropriate vibrations regarding to some molecules. So first they select which laser source they want according to the wavelengths, and at the end it makes the device easier to industrialize in this way as compared to another type of source.
Thank you.
Thank you, Alexander. We had again the same question, but you were first now. Another question for Thierry. I also have a curiosity regarding your solution for measuring the particles or distinguish which particles were in cells were infected and no. Which are the advantages over other techniques like the nanoparticle tracking analysis, other kind of techniques available in the market? I guess you made a solution quite customized for the application itself, right?
Yes, actually, so this is a digital holography, so it has a lot of advantage. First, the best advantage they have is that they can monitor online. That means they don't need to take any biomaterial from the bioreactor. So it is made in a way that the biomaterial is circulated in the focus of the microscope. So it's a really online technology. There is no interaction with the biomedium. So this is the first one. And then this digital holography technique is, well, it's label-free. Of course, that's important. It doesn't need to involve any marker. And it is performing a volumetric analysis, which is quite interesting. And well, so far, that's, and also I think it's a self-calibrated technique. So, well, according to our customer, again, we develop product for customers. So the ultimate advantage they could bring on their competing technology shall better be answered by them. Well, that's the way I can answer to your question.
Mm-hmm. Yeah, good. Thank you. Elizabeth from Hubner.
Thanks, Thierry. Very interesting to hear all your applications. I had a question, the digital holography application there. Do you use lasers for that?
Yes, indeed. Indeed, they use a laser source. I cannot disclose all the detail, but it's well, I don't which type of laser diode or specific source. I cannot tell exactly now. Maybe I could.
Sure. No, I understand. No problem. Yeah. Thanks.
Perhaps in a private conversation later on. Good. So thank you, Thierry. Really nice presentation. And I tell you that I was really amazed when we talked first time and you showed me all the capabilities of Lambda X. So feel free to explore the website and see what can they do for you. And let's move to the next presentation. We are talking about microscopy and illumination. So it's time to see what's new in LED illumination. So I would like to introduce you Gerard Woriske, is the technical director at CoolLED. So Gerard, the floor is yours. And you can share your screen and show us what is new.
Thank you, Antonio. Can you see my screen? Oh, hang on.
Not.
You should. Hopefully now
it's
you
loading
can.
right now. Yeah, it's loading right now. Yeah, now it's perfect.
Thank you very much. So hello, everyone. My name is Gerard Woriske. I'm the technical director at CoolLED. And what do CoolLED do? So we actually design and manufacture high-intensity LED illumination systems for fluorescence microscopy, which can create pictures like you can see on the screen there to help scientists and researchers analyze their cells. So we do this with LED-based systems, and this is a wide field microscopy technique. So it's good for living cells and very fast imaging, at like 100 frames per second or so. We typically sell these to the likes of Olympus, Nikon, Leica, and products like this just fit onto the back of your fluorescence microscope. LEDs really took on bulbs to begin with. The challenge was to take on the mercury metal halide and the xenon bulbs that were the standard or the incumbent light sources in these applications. But there was many benefits for the LED. And over the years, they've become very strong, which was considered their main flaw, to actually really outstrip the bulb on power or radiance and in a bunch of other metrics. Stability is one great feature here. This is over short term and long term. The graph on the right shows a lifetime graph actually, but stability can be very important for ratiometric type work. And with a good current, with good cooling, we can get really great stability in fractions of a percentage in certain circumstances. Long lifetime, the LEDs can be switched on and off, unlike the bulbs, and that can be done very quickly. So lifetimes is generally measured in tens of thousands of hours instead of hundreds or a few thousand hours. The wavelengths of LEDs are discrete and they're narrow, full width half max, typically about 25 to 30 nanometers. This can actually be a benefit for improving signal to noise ratio, although if you've got very good filters, that's not such a problem. But with poorer filters, you have to block out all of the other light you would get from a bulb, which, unlike like an LED, that unneeded spectral power isn't there. Cost effective over time. This is a comparison to a bulb. There may be a higher increase in cost than the original outlay of an LED, but actually cost savings over time are quite significant. Sustainability and finally control. I think one of the best things about being able to control a light source like an LED when you're looking when you're doing bioimaging is that you can significantly reduce the photo bleaching and phototoxicity of your imaging. And the phototoxicity, if you're looking at live cells, can actually affect the validity of your results. If cells start suffering or getting sick, they can affect performance. And the biggest improvement the controllable light sources like LEDs can bring to that is simply to be able to match the LED to the integration time of the camera in microseconds, such that there's minimal, if you like, dead or useless illumination time in either side of the camera exposure. These are a couple of reasons why LEDs are really the go-to solution right now for imaging if you're not using a laser. CoolLED offers a broad range of LEDs. Here we have LEDs we currently use from 280 nanometers all the way to 950 nanometers. We're looking beyond that. So we encourage customers who need a light source. They can choose from any of these. There's even a broad white in there as well. I'll highlight the range of products that we have, and we call it the Amora range, and this is actually aimed at OEM applications, where we allow the customer to choose the spectral coverage that they need. And here irradiance is very important to us. So the method we use is combining LEDs using a dichroic mirror approach, and this preserves the irradiance such that each color is actually adding to the other rather than other mixing approaches. It's a little bit more expensive, but it preserves the irradiance. So these engines, you can choose from anything up to 16 channels, 16 LED channels in one box. And with that, you can customize, we can customize the light derivative for you. We can give you the control you want, and other performance. This is a service we provide. And I think it's important that as well as like a very good optical engine, if you like, or all the light and the optical output that you need, customer support is very important to us. And if you've got a project, as we often sell to sometimes startups, sometimes established companies in high content screening applications or automated diagnostic applications, we can take the worry about the light source away. And just help you through getting the compliance and getting a product that fits in your box that you won't have to worry about. I'm also thinking the EPIC community is going to be filled with people outside of fluorescence microscopy, but maybe still in life sciences, biosciences. And these products are also now finding interest from people doing things like looking at imaging inside the body for checking out cancer, etc., and using fluorescence in that area, in which case we might be coupling to a fiber. Finally, in some new developments, when you think of our LEDs have replaced the bulbs very much in maybe a multispectral imaging application. The bulbs still dominate when it comes to wide unbroken spectral response. One such application area is hyperspectral imaging. Now, putting a lot of LEDs together is simple enough, but actually the trick here is because the LEDs are spectrally different and spatially separate, the trick is being able to get lots of power and homogenize those without using traditional diffusers. And QLED's developed a currently patent pending technique to do this, such that we're finding a lot of interest in hyperspectral imaging right now. And this is a spectrum we've got from our recent prototype that we're showing. You would have seen it in Stuttgart last year, and you'll maybe see it next year as an actual product. But we're improving this spectrum. And what's beautiful with LEDs is you can tailor the spectrum or engineer the spectrum to complement the spectral responsivity of a camera. For example, a typical silicon detector will have very poor sensitivity down in the deep blue and the violet, and again quite poor as it goes up to the NIR. We can turn that, we can create an illumination spectrum to complement that. So I see it as quite an exciting area that we're moving into. And again, there's been lots of interest in the life sciences and hyperspectral, doing things such as maybe, robot arms carrying light sources and cameras over the skin and detecting the condition of moles, for example. But the illumination is a very important part of this. And right now, bulbs are still being used out there. And I think we're still on the journey of replacing those bulbs on those high-end applications. Also, finally, we'll go into this unbroken spectrum from 950 to 1700, but that will probably be next year. So lots we can do with light sources. Very interested in your any questions or any interest in partnering, etc., etc. We've been going in fluorescence since 2006. We've got a relatively small company with just under 50 employees, but we are hiring. We're focused, we're into university partnerships and industrial partnerships to develop cutting edge light sources. So thank you very much. Any questions?
Thank you, Gerard, for this nice summary about your products and your future ideas.
I'll stop sharing.
We have time for questions. I could perhaps start with a short question, and the typical EPIC question: what can EPIC do for you? I guess for your new developments, you will need optical components. Are you missing something in the market, some challenges or needs you see for your future developments in terms of optical elements, fibers, etc.,
Yeah.
etc.?
We use all of those types of things, Antonio. Lenses, dichroics. We've used chroma a lot, actually. Have been very good. Fibers, light guides, the works. We do the design and manufacture here, but we don't create the LEDs and we don't create the optics. We buy all that stuff in. I think that there's an awful lot happening in the industry, and I think where people are involved in light, and if it's not lasers, you're typically looking at bulbs or LEDs. But I think a lot of people aren't quite aware of the where LEDs are going and roughly where they're at right now. Some of the irradiance measurements, coming off the chips about 4 watts per millimeter squared. It's not quite what a laser can do. Actually, grabbing that is actually, we're finding lots of applications still continually coming into reach for the first time. And the life sciences has been a great market for us, and we continue to get lots of interest there. So what can the industry do? It's actually just meeting up, networking, which is easier to do in person rather than on a webinar. But actually just hearing what people's challenges are, and showcasing what we can do as a company is what helps.
Good.
Thank you.
Any other question for Gerard? Well, if not, I thank you again for your nice presentation about all your capabilities. And again, if somebody's interested in being in contact, okay. I see in the chat that we have a question from the YouTube channel from Fabio Caligari. The question is about the medium infrared imaging of biopsies. Is the loss of resolution introduced by that wavelength range a problem for such diagnosis application?
Oh, the loss of resolution in that wavelength range. I'm not sure what he means by that. The mid IR, we're really going into the short wavelength IR up to 1,700. Maybe I've misheard.
Yeah, he's mentioning about medium infrared.
Yeah. Well, we're just conscious that those chips are available, and there is that interest for the I think it's the in-gas cameras that are actually sensitive from about 900 to 1,700. We don't have a product there yet, but we are intending to provide broad spectrum, high radiance illumination there. So sorry, I don't think that's answering your question.
Well, if the person in YouTube is
it gets in touch,
listening, please get in touch with us or with Gerard directly for further discussion. And thank you again, Gerard.
Thank you for listening.
For the presentation of your company. And we are moving to our next speaker is Øystein Hell, from the Chief Technology Officer at Chip Nanoimaging. It was a pleasure to meet him at the last Photonics West. So I'm really curious in what he's going to present today. So Øystein, the floor is yours.
Thank you, Antonio. See. So what can you see now?
Yeah, we see your screen.
All right. Yeah, so thank you for inviting me here. So I'm Øystein, and I will talk about our new method of generating large content optical nanoscopy images for biological applications using photonic chip-based illumination. So let's quickly review what is meant with nanoscopy, which is also known as super resolution microscopy. Some of you, or many of you, might already know this, but the fundamental diffraction limit of light is causing the spatial resolution in a light microscope to be limited down towards 200 to 300 nanometer, which is the resolution in this fluorescence image I'm showing you here. So this is a liver specimen. And here we can see the large dark regions, apparently without any content. So a nanoscopy is then different optical or chemical or mathematical tricks to look beyond the diffraction limit, as most of you already know. For the liver example, this enables the visualization of these nano-sized pores that occupy the earlier dark regions. And this is a set of filters that all of us have inside our liver, which is surrounding the blood vessels in the liver, filtering our blood. The commercial realization of nanoscopy imaging techniques have been around for a few years already. However, there is the current implementation comes with a problem. So the problem stems from the fact that in a conventional system, the specimen is illuminated via an objective lens, and the fluorescence is captured with the same objective lens, typically a high numerical aperture objective lens. And then you have your sample, which is mounted on a piece of glass, a glass slide or cover glass. So this effectively limits the imaged area, causing only a small part of the specimen to be imaged. And the result of this may be that you miss features, or if you need to utilize image stitching, it might cause the imaging technique you're using to be slow. So we have found a solution to this, and we have called it chip-based microscopy. So in a chip-based microscope, we use objective lenses only to collect the fluorescence. And we use a photonic chip to both hold the sample, but also to illuminate it via integrated optical waveguides. So this enables a flat illumination and a free choice of imaging objective lens, and again, a large field of view. If you look at the schematic, how this would look like, we introduce laser light into the high refractive index waveguide on top of the chip. Light is then guided by total internal reflection in the waveguide, and as most of you know, wherever there is a waveguide, there is also going to be a small part that leaks outside of the waveguide in the form of an evanescent field. And that is then exponentially decaying up to around 150 nanometer away from the waveguide surface. And this is what many biologists know as turf illumination. And since we can make very, very wide waveguides, we can also then illuminate very large regions with this type of illumination. The specimen then can be grown directly on the chip surface, the same way as you would do it on glass. And the fluorescently labeled sample would then emit the fluorescent when it comes in contact with the evanescent field. And then you can capture this fluorescence with an upright microscope. So the standard method of doing this, that is the objective-based methods, gives you a very high resolution. It's super resolution, but it is over a very small field of view. Typically 50 micrometers by 50 micrometers, as I show you here. Some have 50 by 80. We chip nanoimaging, using our chip-based technology, we can extend this up to 500 by 500 micrometers, as we show here. So this is then an increase in the field of view with around 100 times. All still in super resolution without any image stitching or anything. Our system consists of a physical instrument. We have chips, we have software, as well as some accessories like service agreements, third-party chemistry, and training. Let's look at a few applications of this technology. So here we see a kidney tissue section, which is imaged using diffraction-limited fluorescence microscopy on a chip. Minimal change disease is due to malfunctioning kidney filtration and cause proteins to be filtered from the blood and ending up in the urine. The term minimal change is due to the nano-sized differences between healthy and diseased samples. So this disease needs to be diagnosed using electron microscopy, which is cumbersome and require difficult sample preparations. So here, the author of this paper, which is cited here, is using chip-based super resolution to reveal the structures used to identify minimal change disease, which is the appearance of these channel-like structures, which is around roughly like 100 nanometer in diameter. Some other examples. These are human placenta tissues used for research on preeclampsia. And there you see an example of a pig heart tissue. So all of these images are in diffraction limited resolution, but in multicolor. Finally, one example of a living specimen. Here, hundreds of cells undergo cell migration. So you can see the cells are moving about on the chip surface. And the nice thing here is then you can get to see so many of them at the same time. And this is useful in the study of wound healing, for example. Yeah, so that was the slides I have prepared. Thank you for the attention. Maybe one more thing, since this is an EPIC meeting, some of the challenges would be nice to address. So maybe one of the challenges for us would be to have waveguide materials that works better on the lower wavelengths down towards 400 nanometer. That would be good for us. Currently, we go down to 450 nanometer. Yeah, and another challenge would be how we could capture even larger fields. We can illuminate almost arbitrarily large fields of view, but we are then limited in camera sensors, pixel size, number of pixels, objectives, tube lenses, and so on. So on the microscope side of the technology. Yes, so thank you very much. I'm open for questions. You can also contact me here on you can see my contact information.
Thank you very much, Øystein. Really nice presentation, and also thank you for sharing your challenges. You have time for questions to Øystein. Yeah, Regina from Tampere University, please.
Thank you very much. Very interesting technique and totally new for me. I have a couple of questions. First one, what's the wavelength you usually use? You said that you need to go down to 400, but what is typical wavelength and how you determine this?
Yes, it's based on fluorescence microscopy. So this typically spans the whole visible range. So right now we have a system working for 450 nanometer and upwards towards the near IR, so upwards like 700 nanometer. But it would be nice to go down to 400 nanometer. A lot of popular fluorophores also in this range.
Okay, and another question, do you understand that it's kind of surface imaging, not deep tissue imaging?
Yeah, so this is a two-dimensional technique, only looking at the stuff that actually goes on the surface of this chip. So this is good for things that are happening in the membranes of cells, for instance, endocytosis, exocytosis, things like that.
Okay, thank you.
Thank you. Lars from DTU, Health Technology.
Yeah. I have a comment to you, Øystein. Thank you very much. This is really cool.
Thanks.
Yeah, because I've been following the work by Stefan Hell.
Yeah,
and I know where all the trouble you have to go through getting the results that he gets. Two things. You probably know that you're banging your head against the Rayleigh scattering in the waveguide, and that probably will make life difficult for you to get to 400 nanometers. But what about 2-photon? I mean, if you use 2-photon excitation, you would be able to ride on the nice dispersion properties in your material, and I'm actually pretty certain that it will work and give you access to the fluorophores excitable in the 400 nanometer region.
No, thank you for that. You're actually the second person who have brought this up over the last week. There was one guy in Photonics West also to comment on that. So thank you. So definitely this is interesting.
You're welcome.
Thank you, Lars. Final question for Øystein? Yes, John. self-question or self-comment?
A kind of comments from a colleague here. We can also do other techniques than the illumination through the chip, so it can be combined with standard illumination techniques through the objective lens, so you kind of can do both or more techniques. Just a short comment.
Thank you for the comment, Jon. And thank you again, Øystein. Really nice technique. As I mentioned, I was really curious, and it's very, very nice. I have another question in YouTube. It's Fabio Calegari again from IIT. How does the chip behave with respect to light polarization?
Yeah, you can send different types of polarization to the chip, and it will work just as fine. There's a little bit difference in how far the evanescent field would penetrate, but it's minimal.
So there is no limitation in terms of the polarization. Do you need to put the light to illuminate the chip?
Well, we use a certain polarization because we have seen that you get a little bit higher excitation for that, yeah. But it will work on other polarizations also.
Okay, good. And the final question, Alexander from Toptica.
Yeah, very cool technique, definitely. I wonder, how do you couple in the light into this thin layer, and how much power do you actually need to achieve a sufficient excitation?
So right now we are using, if you want to use this for super resolution using single molecule localization microscopy, we use 500 milliwatt laser, CV laser. And the method of coupling here is by focusing light onto the end facet of the chip. So we are hitting with a focus spot there. So there is a little bit of loss there, obviously.
Thank you.
Thank you.
Thank you, Alexander. And thank you again, Øystein, for your presentation. And we go last but not least, our next speaker is Maria Frias, System Engineer at SimVivo. And I am very interested in their technology, the OCT catheter imaging for bladder cancer. So, Maria, if you can share your screen.
Yes, thank you. Yes. I hope you see it now in full screen.
It seems loading, probably in a couple of seconds. Yeah, we have it now.
Okay, perfect. My internet is acting out, of course, always Murphy's Law, but just let me know if it's lagging something in the audio. But yeah, welcome everyone. Thank you for your time. And yeah, I'm Maria Fernanda Frias, and I'm working at SimVivo, where we are developing an OCT catheter for bladder cancer imaging. So SimVivo, it's a company that is located in Eindhoven in the Netherlands, and we are working closely together with medical personnel at different clinics and hospitals to develop this catheter. So we have created very small catheters, so it's around 2.5 millimeters outer diameter that works in a standard cystoscope. And our OCT catheter works with our MEMS mirror. This is our main component for the scanning. And as you can see here in the left side, we have our catheter combined with cameras and lights from an already standard cystoscope. But why are we developing this? And yeah, so as Paulo mentioned in the presentation at the beginning of this session, right now for bladder cancer, it's really hard to detect it because current systems through camera, for example, we can only see the surface of the bladder. And it's a very thin organ. We have a wall of around 2 millimeters. And we cannot see how what's the part of the tumors. And current systems such as MRI or CT techniques, they have a low resolution and they cannot help for this. So in order to improve the detection of the tumor cancers, we have developed OCT scanner, which is the optical coherence tomography. And this technique works similar to ultrasound, but instead of using sound waves, we use near-infrared light. And we measured the backscatter light from different layers of the material that composes our sample, in this case, the wall of the bladder. Here on the right side, I have an image of an example of an OCT image of a fingerprint, and you can see the rugosity of the fingertip and then different layers of the skin. So the advantages of the OCT technique is that it's a completely non-invasive imaging technique, and it provides really good in vivo images without damaging the tissue. It has fast scanning rates, and that allows to have visualization in real time. And then we have high resolution, which is very important for such thin layers. We're going in depth in our technology. So we have, as I mentioned before, we have it based on MEMS mirror. So this is a very small component to which we have our light source through the optical fiber that goes through a green lens and then a prism that is then projected to our MEMS mirror. And with this, we start scanning our field of view. We have a field of view of 5 millimeters and a working distance of maximum 11 millimeters. This working distance of 11 millimeters means we can be around 1 centimeter away from the wall of the bladder, and then we can still scan it and go in depth. This is very convenient because, as you can imagine, then it's a very reduced harm for the patient. We don't touch the tissue, so we don't create any extra lesions. Our frame imaging speed, this number has allow us to have a robust system for sorry, I got distracted a bit. So yeah, this frame imaging speed allow us to have a robust system for artifacts in the human body, such as breathing or bowel movements. And yeah, then we can create really nice images. And as I mentioned, we have a very small diameter. We want to provide not only the catheter, but the whole system. So we have the OCT base, where we have our in-house developed software for imaging, so for visualization and image processing. And we also have all the interface for the catheter manipulation. We have our catheter that is, we are planning for now to have it disposable, and we got a sustainability program in the background. And as mentioned, we want to have it as easy as possible integrated into the hospital system. So we have designed it to have it possible to use it with standard cystoscopes, not only because the hospitals already have it, but most of the times it's already positioned in the patient's body. So it's very easy to go in and out with our catheter without disturbing more the process in the OR. Here I show you an example. So this is, we are imaging a pig bladder, a bladder of a pig. And we have worked together with urologists, and then they are very excited to what we are seeing, because it's possible to see blood vessels and different layers of the bladder. For example, you have the fat layer, and then you see the muscle layer, and this is a very nice, promising image. So yeah, we are, as I said, we are working together with them to improve our visualization system, but also our technology. And we are in the process now of ex vivo tests and continue with, yeah, hopefully very soon in vivo trials. But currently we have some challenges that I want to share with you, and our main challenge is that we are miniaturizing optics to fit a small form factor. So we have an external diameter right now of 2.5 millimeters, but we want to go to 2.2. So as you can imagine, for optics, this is very challenging. So we are open to any brilliant ideas that you might have. And then we also want to have a direct tunable laser based on photon integrated circuits. With a target modulation frequency of 400 megahertz. So yeah, these are our main challenges. And then for future applications, we also see some interesting situations. So we would like to have a localization and shape sensing approach with that we will be able to measure with a frequency higher than 25 Hertz for tracking purposes. So this is to position the catheter for location of purposes, so to find the location of previous tumors, for example. And we would like to also have additional modalities. So as you can imagine, this having such a small imaging catheter allows it to go in other parts of the body. So for this, we would like to have different wavelengths to tailor it for the different applications, or for example, Raman spectroscopy, or any other. And we aim to have as an end goal one-stop diagnosed treatment. So we would like to also incorporate treatment lasers, so to, for example, fulgurate the tumor once it's properly imaged. So overall, this is our presentation of the company, our technology, and our challenges. So thanks a lot for your attention, and I'm open for any questions. Thank you.
Thank you, Maria Fernanda. It was really nice, and also thank you for sharing your challenges in terms of miniaturization, different laser needs. So time for question. Yeah, Lars again.
Just a comment, Maria. Your wish for doing laser treatment of bladder cancer can be achieved. Some of my colleagues here in Denmark is doing this at Skype Hospital using the system from Olympus. So Olympus has a system called Soltiv. It's a thulium fiber laser.
Oh, thank you.
The only caveat here is that this is for non-muscle invasive bladder cancer. So there is a limitation to how deep you can go, because the non-invasive bladder cancer is the most easy way to treat. Once it gets beyond that, it's a little bit more tricky. But there is definitely, if you want to team up with people who would love to meet you also, Olympus would be a very good place to start.
Thank you very much. Yes, I would like to have that contact. Thank you.
Thank you, Lars. Any other comment or question to Maria Fernanda? I have a curiosity in terms of miniaturization. You mentioned the optics, but about the detector or the cameras, do you see any need, or are you covered?
So we have our MEMS mirror, that it's our main component. So we don't have like a official camera, let's say, like we know a classic camera. So this is already quite small. So it allows us to reduce the diameter. So this component is not the limiting factor, but the optics around it.
Okay.
Yeah.
Because I was thinking one EPIC member that is called OptaSensor. Perhaps you know them. They are doing also small micro cameras for endoscopes. Just thinking it could be interesting for you. But,
yeah, thank you.
You're welcome. Regina from Tampere.
Yes, thank you very much for the presentation. I have a question regarding laser wavelength. What do you use now?
Yeah, we have a wavelength of 1060 nanometers.
1060. So it's pulse, yes, or CW?
Yeah.
Pulsed.
Yeah.
And another question, how deep can you see now the bladder tissue?
It depends, but we can go up to a bit more than like 800 nanometers, more or less.
Okay, thank you very much.
Yeah.
And Robert from Modulite.
Hello, thanks for a very nice presentation. I would like to ask that with current camera-based detection, it's especially hard to detect these carcinoma in situ tumors because they are really flat. do you think your solution could help with that, or can you see those better with your OCT probe?
Yeah, so our intention is not to remove the camera imaging for the process, but complete it. And the advantage of our technology is that you can penetrate the tissue. So you don't see only the surface, but you can see the layers underneath. So we could see the shape of the tumor, so the penetration of it. And as mentioned by our colleague before, yeah, if the tumor has reached already the lower layers, then it's very difficult. But if we could detect it at the early stages, then it's only the surface level of one of the top layers of the bladder, and then it's very easy to treat. So yeah, the idea is that together with camera images, we can give a better interpretation of the stage of the tumor.
Okay, thanks. It would be really actually beneficial to improve the detection of the KISS tumor if combining this with the camera-based imaging could help with diagnosing also those tumors.
Yeah, exactly.
Thanks.
Yeah, you're welcome.
Thank you, Robert. Do we have any final question for Maria Fernanda? If not, I thank you again for your nice presentation and for sharing your technology with us. I know that we are a little bit over time, but I was really enjoying the discussion today and the presentation, so I hope you also did. Let me just briefly share my screen for the final comments. First of all, thank you again to all our speakers. Thank you to Pablo, Patrick, Jana, Thierry, Gerard, Oysten, and Maria for the nice presentations. Also, thank you to Almut and to Stefan for sharing the technology within the one slides. Again, thank you to our sponsors for supporting us. The topic today, sorry, I'm going back. So thank you to Actar, Chroma, Oxius, Iridium Spectral Technologies, and Huner Photonics for the support to this event. And very briefly about EPIC, you know that we are the European Photonics Industry Consortium. We can help you with the different requests you can have in terms of connecting people, technology needs, access to markets, supporting human resources and investments. So feel free to contact us in case you need help in any of these topics. We have a very fantastic team that are supporting all these activities. And regarding next events, I encourage you to go to our website for our next in-person and online meetings. Some of them will be related with the next OFC conference in San Diego. But I would like also to highlight the first technological meeting hosted by a member. In this case, it's about fiber sensors, hosted by the Portuguese company HBK Fiber Sensing. Will be in April 19th and 20th in Portugal. So springtime in Portugal, very nice city, very nice topic. So I encourage all of you that are interested in the fiber sensing technology to go to our website and register to join the nice in-person discussion and the nice networking with a lot of industrial and engineering end users and scientific partners. And that's all from my side. So I think we can stop the recording.